Hello again and welcome back to the second part of our lecture. So in our first lecture, we've discussed the cell theory and we discussed the classification of prokaryotes, their structures, as well as their genetics. Now in this video, we would like to focus more on eukaryotes, their functions of their different membrane-bound organelles, discuss their cytoskeleton, and then finally go over the modes of the viral replication, discuss prions and viroids, and then as always conclude this video with some practice questions. Alright, so starting with eukaryotes, here I have provided you a summary of all the different structures that you can find inside a eukaryotic cell. And so as you can see, there are so many structures here that have membrane. Like for instance, the lysosome has a membrane, the peroxisome has a membrane, the mitochondria has a double membrane, the nucleus has a double membrane. So actually these are the characteristics that distinguish a eukaryotic cell from a prokaryotic cells, which includes one, presence of the membrane-bound organelles, like here nucleus and mitochondria are example of membrane-bound organelles, and second one is presence of mitotic or meiotic divisions versus binary fission that we see in prokaryotes. So here I would like to discuss the function of different one of these organelles, starting with the nucleus. So here you can see that we have the nucleus that has the double membrane, and then we have the nucleolus that is located inside. So let's start the writing here. So the nucleus is basically the storehouse for the genetic material, where it serves as a storage place for DNA. And then it's inside the nucleus that DNA can undergo replication, or it can undergo transcription, where mRNA will be formed, and then later on this mRNA, with the help of these nuclear pores, can exit the cell and go reach the ribosomes, which are shown here in red, and undergo translation, and by translation proteins will be made. Okay, so going back here, so we just discussed nucleus is the storehouse for DNA. There are histones that will help compact DNA into the condensed form called chromatin. And then during cell division, this chromatin will further undergo coiling and form chromosomes, which is a very condensed form of DNA and proteins. The other function of the nucleus is that it has a nuclear envelope that has pores inside it. So it's basically perforated, and as we just shown it, it's called nuclear pores, and the function of these nuclear pores to allow movement of large molecules. So there are nuclear pores allow movement of large molecules, like for instance mRNA or the ribosomal subunits. So in fact, the fourth function is that inside the nucleus we have nucleolus, which is the location where the ribosomal RNA and ribosomal subunits, as we just discussed, 40S and 60S in eukaryotic cells. So ribosomal subunits are being produced. So rRNA and ribosomal subunits are being produced here in the nucleolus, and then with the help of these nuclear pores, these structures can exit the nucleus, and then once inside the cytoplasm, they could assemble to form the ribosomes, which are shown here in red. Alright, the next structure I would like to discuss is the mitochondria. And so here you can see that the mitochondria is made of a double membrane, and this inner membrane is kind of like finger shaped. It keeps on having like convoluted structures. The reason for that is to increase the surface area. So let's go over that. So the mitochondria is basically a bean-shaped structure, and its job is to supply ATP So we do have a lecture on glycolysis, anaerobic respiration, and aerobic respiration. We have extensively discussed the function of every one of these structures. Complex 1, complex 2, 3, and 4 of the electron transport chain will create a hydrogen gradient, which will later on use by the ATP synthase to generate ATP. And this is called aerobic respiration.